Okay, so today we will be continuing on skeletal muscle contraction. As discussed in the last video, each muscle is made of myocyte. Myocytes are made up of myofibrils and myofibrils are made up of sarcomeres. Now, if we take into account one myocyte as a cell, the cell membrane of the myocyte is sarcolemma. The cytoplasm of the myocyte is known as sarcoplasm, and the endoplasmic reticulum of a myocyte is sarcoplasmic reticulum. Hope this clears the nomenclature. Now, this is what we see is the structure of a single sarcomere. The region of this structure that lies between the two blue lines is one sarcomere. These blue lines are known as Z discs. These purple lines, thick purple lines that we see here are the thick filaments which are made up of myosin, and the orange lines that we see flanking those pink purple lines are the thin filaments made up of actin, troponin, tropomyosin. If we draw a line midway between the structure, we get something called the M line, and we can see that flanking the M line is a B zone where there are only thick filaments. Corresponding to this B zone, there is a I band where you can see there are only thin filaments. We can see another region called A band where there is overlap of thick filament and thin filament. So this is the basic structure of a sarcomere. Now the sarcomere is anchored inside the cell via cytoskeletal proteins. These are of two types, transverse and longitudinal. One of the clinically important transverse cytoskeletal protein is dystrophin, and we know its implication in Duchenne muscular dystrophy. Dystrophin is an actin binding cytoskeletal protein which anchors the sarcomere to the cell. The longitudinal cytoskeletal proteins are titin, nebulin, and alpha activin. Titin, we can see, are these red lines which help to give structural integrity to the myosin heavy chains, and they help in anchoring the myosin heavy chains to the Z disc. Nebulins are the pink lines we see inside the orange actin filaments, so they give structural integrity to the actin filaments, and alpha activin help in anchoring the actin filaments to the Z disc. Titins are important in cardiomyopathies. Now, just a minute, we'll pull up another picture. For a skeletal muscle contraction to happen, there are a temporal sequence of events that happen. First of all, there is action potential generation. This is followed by an increase in intracellular calcium concentration, and then there is skeletal muscle contraction. This is the temporal sequence of events that lead to skeletal muscle contraction. So there is excitation via action potential. This causes calcium increase inside the cell, and this causes contraction. This is known as excitation, contraction, coupling. Now, how is action potential generated? So this is what we see is a neuromuscular junction. This is a presynaptic neuron, this is a postsynaptic neuron, and this is the synaptic cleft. Once action potential is, there is generation of electricity in the presynaptic neuron, the calcium channels open in the presynaptic neurons, and these calcium channels bring the calcium inside this presynaptic neuron, and this causes the release of acetylcholine containing vesicles into the synaptic cleft. This acetylcholine now binds to ligand-gated ion channels on the postsynaptic neurons. These channels are also sodium-potassium ion channels, and they bring sodium inside the cell and potassium out of the cell, because sodium is primarily an extracellular cation and potassium is primarily an intracellular cation. This movement of sodium and potassium leads to generation of action potential. Now, once the action potential is generated, we can see its effect on a single myocyte. This action potential now traverses down a transverse tubule which runs along the sarcolemma. This is the transverse tubule. Now, once action potential enters into the transverse tubule, it depolarizes the dihydropyridine receptors present on the T tubules. These are L-type voltage-gated calcium channels. The depolarization of the dihydropyridine receptors causes opening of the ryanodine receptors on sarcoplasmic reticulum. So this is what we are seeing is a myocyte. This is sarcolemma, this is sarcoplasm, this is sarcoplasmic reticulum. Action potential first entered. It causes depolarization of the dihydropyridine receptors on the T tubules. This depolarization causes opening of the ryanodine receptors on sarcoplasmic reticulum. The sarcoplasmic reticulum actually is a calcium reservoir inside the myocyte. Once the ryanodine receptors open, there is inflow of the calcium from the sarcoplasmic reticulum into the sarcoplasm. Calcium is primarily an extracellular ion. Opening of the ryanodine receptors bring calcium into the cell. Now, this calcium will go and bind to the C-type troponin. We remember that thin filament is made up of actin, tropomyosin and troponin. These are actin, this is tropomyosin and this is troponin. Each troponin has I, T and C components. This calcium binds to troponin C and causes a conformational change in the tropomyosin orientation, and this tropomyosin now is the tropomyosin is now removed from actin. What happens in a relaxed state is that the tropomyosin covers the myosin binding sites of actin and prevents the binding of myosin to actin and helps to keep the muscle in a relaxed state. Once calcium binds to troponin C, there is a conformational change and this tropomyosin is removed from the actin and the myosin binding sites of actin are now opened up. Now the myosin can bind to actin. So what happens next is what is known as, I'll just pull up a picture here. It is known as the cross bridge cycle. Now we have already reached a state where the myosin has bound to actin. Here we can see myosin is bound to actin and this causes a state of initial contraction. In a living cell, this first contraction is short-lived. Once there is ATP in the picture, ATP comes and binds to the myosin head and this releases myosin from the initial binding site of actin. This causes conformational change and the myosin head tilts and moves forward a little towards the positive end of actin. Now the ATP is hydrolyzed to ADP and this myosin head moves towards the positive end and binds to the next actin protein. So we can see that the myosin moved from here to here. This is known as from here to here, from here to here. This is known as myosin walk along the actin filament towards the positive end. And we can see that this was brought about by the presence of ATP. In a dead cell, when there is no ATP, the initial binding of myosin to actin is sustained and this sustained state of contraction is known as rigor. Once the contraction has happened, then this state of excitation contraction coupling will continue to happen till the action potential is present. Once it's gone, the sarcoplasmic reticulum, which is the calcium reservoir of the myocyte, takes up the calcium from the cell via sarcoplasmic endoplasmic reticulum calcium ATPase channel. So now the intracellular calcium level falls and there is muscle relaxation. Inside the sarcoplasmic reticulum, this calcium is bound to a protein called calsequestrin. So this is the sequence of events that lead to skeletal muscle contraction. We shall discuss smooth muscle contraction in our next tutorial.